lapel.
Learning her principles of biochemistry. Chapter 1: The Foundations of Biochemistry. Biomolecules are compounds of carbon with a variety of functional groups. Welcome back to our biochemistry crash course. Today, we're diving into the fascinating world of biomolecules and their role in living organisms. Let's talk about carbon. It's the unsung hero of biochemistry. Why, you ask? Well, it's all about carbon's bonding versatility. You see, carbon can form a variety of bonds with other elements, making it the perfect building block for the molecular machinery of cells. Now, let's talk about functional groups. These are the specific arrangements of atoms within a molecule that give it unique chemical properties. We've got hydroxyl groups, amino groups, carbonyl groups, and so much more. These functional groups are what give biomolecules their distinct biological and chemical personalities. Moving on to macromolecules. These are the heavyweights of the biomolecule world. We're talking about proteins, nucleic acids, and polysaccharides. They're like the rock stars of the cellular stage, with each type playing a crucial role in the functioning of living organisms. Now, let's get into stereochemistry. Biomolecules often exist as stereoisomers, meaning they have the same chemical formula but different spatial arrangements. This can have a big impact on their biological activity, as we'll see later on. And finally, we can't forget about the molecular components of a cell. From proteins to nucleic acids, lipids to polysaccharides, each biomolecule plays a unique role within a cell. Cells contain a universal set of small molecules. Cells are the building blocks of life, and they all share some key features. They are bounded by a plasma membrane, contain a variety of small molecules, and have a set of genes that hold the instructions for the cell's functions. Transition to an animated depiction of the chemical structure of small molecules. Now, let's talk about the small molecules present in cells. These molecules are the essential players in the intricate dance of life. They include metabolites, coenzymes, inorganic ions, and enzymes, all working together to keep the cell functioning. These small molecules are incredibly diverse and play crucial roles in various cellular processes. From energy production to signaling and structural support, they are the unsung heroes that keep the show running behind the scenes. When we look closely, we find that the collection of small molecules in a cell carries out a program, ensuring the cell's survival and ability to reproduce. This intricate dance of molecules is the very essence of life. These small molecules are not unique to a specific organism. They form a universal set found in all living organisms, from simple microorganisms to complex multicellular beings. The story of these small molecules goes back billions of years, to the very origins of life on Earth. As life evolved, these molecules became the foundation upon which all living organisms were built. Macromolecules are the major constituents of cells. So, what are macromolecules? Well, they're large, complex molecules made up of smaller subunits called monomers. These macromolecules are essential for the structure and function of cells, and they include proteins, nucleic acids, carbohydrates, and lipids. Proteins are made up of amino acid subunits and play a crucial role in a wide range of cellular functions, from structural support to enzymatic activity. Nucleic acids, such as DNA and RNA, store and transmit genetic information, allowing cells to carry out essential processes like replication and protein synthesis. Carbohydrates serve as energy sources and structural components in cells, while lipids are involved in energy storage and cell membrane structure. Together, these macromolecules make up the building blocks of life. Now, when we talk about macromolecules, we're also talking about the intricate processes that occur within cells. For example, protein synthesis involves the complex assembly of amino acids to form functional proteins, while DNA replication and RNA transcription are crucial for genetic information transfer and gene expression. Understanding macromolecules is key to understanding the fundamental processes that make life possible. From providing energy to maintaining cell structure, these large molecules are the backbone of all living organisms. So, the next time you think about cells and their functions, remember that it's all about the macromolecules working together to keep things running smoothly. Box 1-to-1 molecular weight, molecular mass, and their correct units. In biochemistry, understanding molecular weight and mass is crucial. Let's start with some key terminologies. Molecular weight is the ratio of the mass of a molecule of that substance to 1/12 the mass of an atom of carbon-12. It's a dimensionless ratio with no associated units. On the other hand, molecular mass, denoted as m, is expressed in daltons, da. 1 dalton is equivalent to 1/12 the mass of an atom of carbon-12. The atomic mass unit, U, is another convenient unit for describing the mass of a single atom or molecule. It's defined as 1/12 the mass of an atom of carbon-12. Let's put this into perspective. Consider a molecule with a mass 1,000 times that of water. We can express it as equals 18,000 or m equals 18,000 daltons. We can also describe it as an 18 keta molecule. However, it's important to note that the expression equals 18,000 U is incorrect. Three-dimensional structure is described by configuration and conformation. In biochemistry, the three-dimensional structure of biomolecules is super important. It's all about how the molecules are arranged in space and how they interact with each other. Let's talk about configuration and conformation. Configuration refers to the fixed spatial arrangement of atoms in a biomolecule, while conformation is the specific orientation of the molecule's constituent atoms in three-dimensional space. Biomolecules are made up of polymers of simple subunits, like proteins, nucleic acids, and polysaccharides. These molecules have large numbers of monomeric subunits and high molecular weights. When these biomolecules interact, the fit between them is often stereochemically correct, meaning they are complementary in their structure, which is essential for their biological interactions. But how do these biomolecules achieve their specific shapes? Well, it's all about the arrangement of the molecule's constituent atoms in three-dimensional space, and it's determined by the specific sequence of monomeric subunits. So, the three-dimensional structure of biomolecules is crucial for their function in living organisms. It's all about how they're configured and how they interact with other molecules. Box 1-2 Louis Pasteur and optical activity, in vino veritas. Louis Pasteur, a prominent scientist in the 19th century, made significant contributions to the field of biochemistry. One of his most notable discoveries was related to optical activity, a concept that revolutionized our understanding of molecular structure. Let's understand the concept of stereoisomers. These are molecules with the same chemical formula and connectivity, but they differ in the spatial arrangement of their atoms. In the case of tartaric acid, Pasteur separated crystals of two stereoisomers and showed that they rotated plane polarized light to the same extent but in opposite directions. This phenomenon is known as optical activity, and it arises from the asymmetric arrangement of atoms in the molecule. When a carbon atom has 4 different substituent groups, it is called a chiral center, and the molecule becomes optically active. To name stereoisomers, biochemists use the RS system, where each group attached to a chiral carbon is assigned a priority. If the priorities decrease in a clockwise order, the configuration is labeled as R, and if they decrease in a counterclockwise order, it's labeled as S. Another naming system for stereoisomers is the D&L system, which is based on the spatial arrangement of the molecule. This naming system is crucial in describing the stereochemistry of biomolecules. Louis Pasteur's work on optical activity laid the foundation for understanding the three-dimensional structure of biomolecules and their interactions in biological systems. Interactions between biomolecules are stereospecific. Chirality refers to the asymmetry of a molecule, particularly when it contains a carbon atom with 4 different substituents. These chiral carbon atoms give rise to stereoisomers, which are molecules with the same chemical formula and bonds, but different three-dimensional arrangements. Enantiomers are a type of stereoisomer that are mirror images of each other, similar to our left and right hands. On the other hand, diastereomers are stereoisomers that are not mirror images of each other. These unique properties of stereoisomers have significant implications in the interactions between biomolecules. One of the characteristics of enantiomers is their optical activity. In solution, enantiomers can rotate plane polarized light in opposite directions, a property known as optical rotation. This distinct physical property allows us to differentiate between enantiomers. The three-dimensional structure of biomolecules, such as proteins and nucleic acids, plays a vital role in their ability to interact with other molecules. The complementary fit between biomolecules is often stereospecific, meaning that the specific arrangement of atoms in space is essential for the interaction to occur. Thank you for watching. Don't forget to like, share, and subscribe for more content on biochemistry and molecular biology. See you in the next video.
Pharmapal.